I think a lot of this will be covered in the upcoming talk, which is much awaited, about the local superfoods and the gut health by Dr. Balamurgan Ramdas, who's extensively working in the gut microbiome related topics, right from mental health to cancers to anemia and to nutritional deficiencies all over the country. And this session would be chaired by Dr. Vishwaranjan Mishra, who is the additional professor, Department of Psychiatry of AIIMS Bhubaneswar. And as I said, the speaker, Dr. Balamurgan Ramdas, is the head of the Center of Excellence for Clinical Microbiome Research. And it's a wonderful opportunity to hear from him, who also happens to be from the Department of Biochemistry, AIIMS Bhubaneswar. So over to you, Dr. Mishra and Dr. Ramdas.
So I should thank you for organizing this event. I'll just introduce the speaker, Dr. Balamurgan Ramdas. He has been associated, particularly, he has been working on the gut microbiota, and he's heading the Center of Excellence for Clinical Microbiome Research. And we know that the link between the brain and the brain-gut axis is a well-established entity, and the implications of different psychiatric disorders, the role of psychiatric disorders manifesting in different functional bowel disorders is already established. The role of gut microbiota is already established in depression and alcohol use disorders. So it is an area of interest for all of us. With these words, I would request Dr. Balamurgan to please enlighten about his topic on local foodstuff and the gut health. Over to you, sir. Thank you.
Thank you, Dr. Vishwaranjan Mishra. Thanks, Dr. Sri, and the organizing committee for inviting me to talk. I'm sure the picture that you see in this slide is a common household picture of in India. And I'm at least sure that there's this folklore that people would have heard, that Krishna stealing butter. So the butter is one of those superfoods that are available, that is commonly available around us. And there are many more such similar superfoods that are available around us. So I will just, today in the talk, I will just go over certain common foods and its implication on gut health. So I just added one more sentence to the given topic. So nothing, it's just the implication of the superfood on the healthy gut microbiome that is present within and on us, and how the superfood actually gives us an opportunity to intervene this gut microbiome. So India typically is a diverse country in many ways, and one of the ways we are more diverse is the dietary habits. Across the country, we have different food habits, and if you pay attention to the food that we eat, the common features across the country is the different spices that we eat and the fermented foods that are prepared in our house. As opposed to the West, we make our own yogurt at home. The dahi or the curd that we call is commonly made at everybody's house. And then there are a lot of other vegetables and fruits that are added to our daily plate. Those are prebiotic in nature. Either they are prebiotic, postbiotic, or probiotics. These are certain common signatures of the Indian diet. Here are a few examples. If you see this Amul probiotic dahi, that's the one you buy from the shop nowadays. Otherwise, we make this curd at home. I make curd at home. And then this curd is a good mixture of probiotic and postbiotic. A probiotic is nothing but a live organism that are known as beneficial bacteria. Okay, these are live organisms. Whereas postbiotic is the product or the metabolites that these probiotics drop into this food items. So the fact that you see in the curd is the postbiotic. Essentially, the food that curd, or the next picture next to the curd you see is the pokal bath, which is the rice plus the fermented rice water. You see these two have good mixture of probiotics and postbiotics. You see on the down panel, these are called prebiotics. In South, they call it sundal, while here, these are the pulses that are steam cooked pulses. The raw banana, which is fried in multiple ways and consumed. And then there is the sweet potatoes. These are high in prebiotic content. And a prebiotic is nothing but a complex carbohydrate. Either takes a lot of time to get digested by the enzymes that we produce, or it can be only digested by the bacteria that are residing in the gut. So which means we are throwing food for the bacteria that are living within the large intestine. The postbiotic you see in the right corner panel is the butter. Butter is highly rich in short-chain fatty acid. And this is a byproduct of bacterial metabolism. So let me just throw light on these, how these postbiotics formed. So how they formed and how do they act? You see in the right corner, there's this carbohydrates or the complex carbohydrates. These are polysaccharides. They can be digested by us or not digestible by us. These complex carbohydrates reach the large bowel, and then they are fermented by the microbes that are residing there. This is equivalent to making curd, where you just use a starter culture and then add the milk, which contains lactose and other oligosaccharides. These feed the lactobacilli that is present in the starter culture, and then they ferment the lactose and the oligosaccharides that are present in the milk, and then they produce the predominantly butyrate, acetate, and propionate. And these are the major content of your curd and other fermented products. And this is exactly happened in our gut as well, where the end product are butyrate, acetate, propionate, hydrogen gas, methane gas, and carbon dioxide. These are all that are formed in the large bowel. And these short-chain fatty acids are further recognized by the GPR43, GPR41, GPR109A receptors that are on the epithelial surface. Or they are also some proportion of them also get readily absorbed or transported across other transporters that are present on the epithelium. These short-chain fatty acids are also readily taken up by all cells across our body. So whichever surface it comes in contact with, it gets absorbed, and then it overexpresses this receptor based on the short-chain fatty acid that is more predominant. Like GPR109A is more overexpression is due to the butyrate that is present in the environment, or it will be GPR43 if it is acetate or propionate. Okay, and so the complex carbohydrate, like the raw banana that we eat, reaches the gut, And then these are worked on by the gut microbiome. And then they produce this short chain fatty acid, and then the short chain fatty acid provides the various health outcomes. The health outcomes that is mentioned in the bottom panel is all about the immune response, but it not essentially works on the immune response. It also does other beneficial functions like absorption of micronutrients and so on. If you see the POCHAL panel, it is at the level of butyric acetate and propionate and the butter is also put on the straight in the region where there's this short chain fatty acid, because they are highly rich in these short chain fatty acids. All right. So now we know that there is this microbiome, and then what do they actually do? Microbiome is implicated in almost all the functions that occur in human body now. It's implicated in Alzheimer's to Crohn's disease to any disease that you can think of nowadays. So there are plenty of correlative studies, as well as there are accumulating cause or effect studies. So what do they offer us? They offer not only the immunomodulatory effects that I mentioned in the previous panel, the microbiome also helps in providing or assimilating micronutrients. It also helps in synthesizing certain vitamins like B12 and K. It also helps in various reduction of various local inflammatory markers. And there are certain in utero benefits that we have due to those epigenetic imprinting in the tissues. So these are certain benefits that they offer. So what is this microbiome? Microbiome is nothing but sum total of all the microbes and their genetic material. So this will give a list of microbes that are present or possibly present in a particular niche. If we talked about gut microbiome, then this is nothing but the list of microbes that were present in the gut. So a microbiome study essentially involves understanding who they are. So basically we take stock, and then we also try and understand what they do. So basically the structure function of the microbiome in a particular niche. And the third important aspect in terms of microbiome is how stable they are. So these three factors actually determine health of an individual. If a microbiome structure function and stable, then individual is healthy. So the factors that actually influence are the age, gender, geography, host physiology, lifestyle, and host genetics. And the disruptors on a regular basis are the diet, infection, and the treatment associated with infection. And I also have what is called SES, the socioeconomic status, that again depends on the exposure to diet and infection. So these are certain determinants of a healthy microbiome. So what do we do here in the Center of Excellence for Clinical Microbiome Research? We have various people working with at least 7 different specialties together, and we are open to include multiple folks across this institute and outside. So we do metagenomic studies. We are about to start the metabonomic studies and metatranscriptomic studies. So basically, we try and understand what they do and who they are and what they do. So basically, we make a predictive tool for various diseases. And all this are out of my experience. And then what I'm about to share are some of my publications or certain ongoing studies. So I'm not including anything that is out of my lab or my work that I have done in the past. So if you see this slide, we have done an earlier study to characterize the fermented food, the homemade curd. We call it in southern India, but this is applicable to most of the curd that is made at our home. So the graph that you see in the bottom panel, where there is an inverted V, is nothing but a peak where you find a maximum amount of lactic acid bacteria. So which means at the 12th hour fermentation, you find maximum amount of lactic acid bacteria. So the lactobacillus, for example, is the kind of bacteria that predominates at the 12th hour of fermentation in the homemade curd, which means we ferment this milk overnight with the starter culture, and then we consume this curd for lunch, or people add it to their fermented rice water in the morning. So basically, we consume good amount of lactobacilli, or the lactic acid bacteria put together from the homemade curd. If you looked at the second panel, where there's multiple colored bars, these are the antimicrobial activity of this strains that we isolated from this homemade curd. The red bars that stand are the ones that are against Salmonella typhimurium. So the various red bars are from various lactobacilli strains that we isolated from this curd. So if you see, most of this lactobacilli strains are produce maximum antimicrobial activity against S. typhimurium. The other colored ones are Vibrio cholerae and Shigella flexneri and E. coli are the other bacteria that we have worked on. And if you see the panels on the side, we found that these are like co-cultured experiments in cell culture, where we co-cultured this bacteria with cell lines, and then we estimated the amount of TNF alpha, IL-12, and IL-10 cytokines that are expressed. And then we find that certain lactobacilli strain produce the anti-inflammatory cytokine IL-10, while most of them trigger some amount of TNF alpha. So they're all immunogenic. The next study, these are all published studies. So next study is that we use what is called dietary resistant starch from maize, from the cornstarch. So we use the dietary resistant starch, and then we make cookies. If you see the bottom panel, this girl has this choco chip cookie with her, and this is spiked with the resistant starch. So what we did is we took two groups of children, and one is healthy children, and the other arm is stunted children, and then we fed them with this resistant starch, and then we looked at the production of short chain fatty acid, acetate and bitrate, and then across a period of time, and then we found that there is this increase in acetate and bitrate levels in healthy children when compared to the stunted children. And we looked at microbiome, which is not published. And then in this particular study, the follow-up study was done in Malawi, where we looked at the zinc role of the short chain fatty acid in zinc absorption. So that is not published yet. So that's data is analyzed. And then, so to establish the role of short chain fatty acid in absorption of micronutrients. Right. So these are some of my ongoing studies where we are feeding the same resistant starch at a higher concentration to HIV positive individuals. And then if you look at this panel on the left, you see we fed this HIV positive individuals for about every 15 days. We changed the resistant starch to either a washout period or a regular starch. And then we found the regular cornstarch that is available in the market. The brand we used is Ruchi, so I'm not marketing for Ruchi, but I'm just letting you guys know because there is a reason that why I'm letting you know. I'll let you know why. So if you looked at the panel, the blue colored lines are the dynamic movement of microbiome from one time point to the other time point, which means one feed to the other feed. So it was not resting at one time point. We have managed to shift the gut microbiome from S1 all the way. S1 is right here and all the way to S9 at the end of the study, which is here, which is somewhere here. And the trajectory is a haphazard trajectory. And then it's an ongoing study. We have measured CD4 counts. We have measured short chain fatty acid. We have measured the viral load at each of these time points. And then we are analyzing the correlation between change in microbiome versus the short chain fatty acid production versus all the immune response versus the viral load that's present in these HIV individuals. So here you see the multiple colored pattern. These are top 10 microbes that are there in each of those time points. And you see that over a period of time, this is 2 patients that I'm showing. Over a period of time, the colors or the patterns don't look similar. So that is the dynamic trajectory that you see on the left side panel. The next ongoing study is what is called a NIOBA study. A NIOBA is nothing but native yogurt banana shake study. What we are doing here is this is an intergenerational study where we are feeding the pregnant women and expecting to intervene early childhood malnutrition. What you see here is we suspect that there is a lot of empirical iron supplementation that happens during pregnancy. This study is an ongoing study where Dr. Samrendra is a part of my study in this. And then we have also proposed a larger study with this, where we suspect that this empirical iron supplementation and has an implication on the gut microbiome of the pregnant women. Pregnancy is already a dynamic in terms of microbiome and immune responses. And on top of that, we add this empirical iron supplementation and we add more complexity to this dynamic situation. So we are trying to intervene this by supplementing the pregnant women with NIOBA for 6 months, about 180 days. A field worker goes door to door and feeds them with a NIOBA, and then we give them the iron tablet, and then we give them the NIOBA, and then we are looking at the iron status in these pregnant women. And here on the left bottom panel, you see that it's a preliminary data. It's also published in IGMR, where we've shown that the male and female take a different path in terms of microbiome. So we suspect that that this female pattern has got some kind of evolutionary adaptation to carry out some special functions, of which we are working on now. Am I running late or I'm on time? Okay. So the other studies that are near completion is another intergenerational prebiotic feeding study where we were asked to look at the pathogen levels and the short chain fatty acid levels in children and women separately. And then, so we looked at, after feeding this prebiotic. So all the red bars that you see are post-feeding increase in the short-chain fatty acids. And then the green bar that you see is increase in lactobacilli after feeding these children for about 6 weeks. Finally, we have been awarded the Gates Grand Challenge for fermented food. This is a study that we will undertake with INCLIN, and I'm sure you heard a talk by Dr. Manoj Das. And we are one of the 5 teams that are selected to be funded. The last one you see is the Pokala, the fermented rice water in India. All the other 4 teams are from outside the country. And in this study, we are planning to extensively characterize fermented rice water and its implication in mother and child health. So to sum it up all, dietary habits have an implication on the microbiome. Short-term implications can be reset, while long-term dietary habits have implications which is kind of debatable, where people talk about enterotypes. There are only 3 enterotypes like the blood group that are available, but those are debatable. Those are debatable results that we have. While from our experience, if you see the panel here, there's the green color and the red color panel. The green color is from the tribal population that we studied, and it's published data. So the green color is from the tribal population, from the kind of dietary habits they have. We have the green colored pattern predominance, while the nearest rural population, which are different in their dietary habit, have what is called an actinobacteria predominance. So, so I think this long-term diet, especially the Indian diet, has a detrimental effect on the gut microbiome and health. Thank you for the opportunity again, and then I have a few acknowledgements to make. Thanks again. I'm open for questions.
Thank you, Dr. Bala, for a comprehensive presentation and opening up a new, whole new chapter towards how gut microbiome is related to mental health and how important it is to improvise the nutritional aspect of women and children.
Mention the de-addiction and microbiome.
Yes, so fortunately, the Department of Psychiatry is also working on the gut microbiome, especially the addiction aspects and how the behavioral changes or the behavioral impacts of continuing the addictive behavior is linked to the gut microbiome.